Yes, good evening, Harsh. Good evening, Karan. Good evening, Ashish. Yes, Chirag. Good evening, beta. Good evening. Yeah, good evening, good evening. I hope the audio and visuals are perfectly fine. Yes, beta, I'm discussing about anaerobic glycolysis and citric acid cycle. I would be happier if you guys will show interest in this particular topic, except for the date going to Kyrgyzstan or no, some picnic spot over here right now. So yes, yes. Shehzad, very good बेटा. Manish, very good. Yes. हर्ष, ठीक है भैया. चले जाओ, पहले तुम ही लोग चले जाओ, हम बाद में आ जाएंगे. चलो, ठीक, ठीक है, ठीक है. चलो बेटा. चलो, 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 चलो. Now, when I say anaerobic glycolysis, beta, firstly, just try to learn the difference. Okay. When I say aerobic glycolysis, aerobic glycolysis. So aerobic glycolysis, beta, yes. Good evening, beta Makpool. Good evening, beta. Welcome to the session on aerobic glycolysis, anaerobic glycolysis, and I will be discussing citric acid cycle. So let me just give you a basic difference between aerobic glycolysis and anaerobic glycolysis. See, beta, aerobic glycolysis, the presence of, it occurs in the presence of oxygen and mitochondria, right? And mitochondria. And when I say anaerobic glycolysis, remember, beta, it takes place in, yeah, yes, good evening, good evening. Yeah, it takes place in absence of oxygen or mitochondria can be absent, or mitochondria can be absent. If oxygen is present, mitochondria is absent, then also anaerobic glycolysis will take place. Then also anaerobic glycolysis will take place. So yes, I hope everyone has gone through yesterday's lecture where I have discussed the aerobic glycolysis. Yes, Asjad Raja, good evening, beta. So just give me a thumbs up if you have gone through yesterday's lecture, beta. So I will be asking you regarding, yeah, I hope you have gone through yesterday's lecture. So I want everyone to give me a thumbs up in the chat box if you know about the glycolysis, the aerobic glycolysis, the steps. So yes, Kriti, Kiran, very good, beta, very good. So I will be using this, yes, steps over here, beta. I will be using these steps over here. So yes, let me use another slide. Let me use another slide. Yeah. Chalo, very good, Kriti, Kiran, Shehzad, Harsh, Ashish. Perfect, perfect, beta. Now let us focus on anaerobic glycolysis. Yeah. And today you guys are going to tell me because anaerobic glycolysis and glycolysis is mostly same. So yes. Now the first product that it will be started with, it will be glucose. Glucose. Now glucose will get converted to glucose 6 phosphate. You already know it will be get converted to glucose 6 phosphate. Now I want everyone to answer the enzyme which will help in the conversion of glucose to glucose 6 phosphate. चलो फटाफट से आंसर बता दो. You already know glucose is a 6 carbon compound. Okay, we have 6 carbon, and on the 6th carbon basically what is there? Now phosphate is coming. Now phosphate is coming. Yes, very good, Kriti, very good, Ashish, Rajvardhan, Ash, Harsh. Perfect, perfect, beta. It is the hexokinase. I have already discussed it yesterday, right? I have already discussed it yesterday, and yes, this phosphate, it is coming from where, beta? It is coming from ATP. ATP is breaking down. Okay, the ATP is breaking down to form adenosine diphosphate. एंड देखो ट्राइफॉस्फेट था, ठीक है. एक फॉस्फेट कम हुआ तो वो कहां आकर ग्लूकोज के 6th कार्बन के साथ जाके लग गया. ओके वो ग्लूकोज के 6th कार्बन के साथ जाके लग गया, सो इट इज ग्लूकोज 6 फॉस्फेट. And you already know, okay, which ion is required in this particular type of reaction. Which ion, I want everyone to answer this question, which ion is responsible in this reaction. And yes, Harsh, very good, it is the magnesium ion. मुझे बहुत खुशी होती है if you guys study and know, come and give me answers. It's like I feel very very good. And can anyone tell me, beta, is it reversible or irreversible reaction? Rai, Naveed, Ashish, Kriti, Rajvardhan, Kaleem, Kiran, very good, beta. Now, is this reversible or irreversible reaction, beta? Yes, it is reversible or irreversible reaction, beta. You already know the enzyme over here. It will be hexokinase. Hexokinase. Yeah, hexokinase. Or we can have glucokinase over here. Right. So I'll just, yeah. Yes, it is irreversible. Very good. Kriti, Rajvardhan, Ashish, Kiran, Harsh, Ash, Kaleem. Very good, beta. It is an irreversible reaction. Now, from glucose 6 phosphate, beta, what will be formed? Yes, it will be fructose 6 phosphate. It will be fructose 6 phosphate. Now, I already told you glucose 6 phosphate and fructose 6 phosphate. These are structural isomers, functional isomers are there. Okay. Glucose का भी structure same है, fructose का भी structure same है. Okay, they have the same molecular formula but different structure. Okay, they have the same molecular formula but different structure. Which enzyme will be here, beta? Which enzyme, okay, will be used over here? Tell me. Yes, very good, Harsh, very good. It is isomerase. Yes, it is isomerase. I'm not like yes, you already know it is the isomerase, beta. Isomerase. So already we have discussed this yesterday. I'm just giving you a brief like yes, phosphohexose isomerase है, ठीक है, ये कल अगर बता दिया गया है तो let us just continue. Yeah, from fructose, yeah, from fructose 6 phosphate, what will be formed? Fructose 1,6 biphosphate. Up. Now, one more phosphate will be added, beta. One more phosphate will be added to which carbon? First carbon. To which carbon? First carbon, right? One more phosphate will be added to, yeah, the first carbon. And you already know the enzyme which will be used over here. Yes, tell me, which enzyme will be used over here? Which enzyme will be used over here? You already know, yeah, that phosphate, it is coming from the use of ATP and it is converting into ATP. Magnesium 2 plus is required. Again, this will be hexokinase, okay, or phosphofructokinase. Better write it like this. Yeah, it will be kinase only. Yes, everyone is right. Everyone is right. Phosphofructokinase है. ये मैंने कल भी आपको बता दिया है. PFK1. PFK1. Now this fructose 1,6 biphosphate, beta. This fructose 1,6 biphosphate, it will split into two parts. Okay, it will split into two parts. Okay, it will split into two parts. Yes, it is the rate limiting enzyme. Very good, Harsh. Very good, beta. It is the rate limiting enzyme. Fructose 1,6 biphosphate, beta. It will split into, मैंने बताया था दो मेजर कार्बोहाइड्रेट्स जो तीन तीन कार्बन के बने हुए हैं. ग्लिसरेल्डिहाइड, yeah, 3 phosphate, right? Glyceraldehyde 3 phosphate, and we have dihydroxy acetone, yeah, dihydroxy acetone phosphate. Okay, dihydroxy acetone phosphate. कुछ नहीं है, इसमें भी तीन कार्बन है. थर्ड पोजीशन contains phosphate. Over here also the third position will contain the phosphate. Okay, these are two carbohydrates attached with the phosphate group. These are the two carbohydrates that are attached with the phosphate group. Now beta, I am just shifting to another slide. You will continue in the same slide, beta. What is the difference in the aerobic and anaerobic? You guys are going to tell me today, right? So you will continue in the same slide. Okay, all the reactions, all the enzymes are same. Now, wherever will be the change, I will tell you. Now, you already know we have, yeah, dihydroxy acetone phosphate, and we have glyceraldehyde phosphate, glyceraldehyde 3 phosphate. Yes. Now, this dihydroxyacetone phosphate, okay, glyceraldehyde 3 phosphate, okay. Now, this dihydroxyacetone phosphate will get converted. बेटा ये भी 3 कार्बन का बना हुआ है. Okay, it is also composed of 3 carbon. Okay, it is also composed of 3 carbons. We have the same, yeah, same molecular structure is there, but different, yeah. So which enzyme will be used beta over here for the conversion of dihydroxyacetone phosphate, okay, to glyceraldehyde 3 phosphate? Which enzyme will be used beta? मैं मारूंगा अगर तुमने काइनेस बोला तो. हाँ, बता दो, बेसिक तो बता दो, इतना तो पता ही होगा. या इट इज द phosphotrioisomerase. वेरी गुड राजवर्धन, इट इज द आइसोमरेज बेटा. वेरी गुड, आइसोमरेज यूज होगा ना. या ये आइसोमरेज होगा. यस, वेरी गुड. फॉस्फोट्रायो आइसोमरेज लिखा हुआ है, कोई दिक्कत नहीं है. फॉस्फो ट्रायो आइसोमरेज कर दो. ट्रायो मतलब 3 कार्बन है, फॉस्फेट ग्रुप लगा हुआ है, 3 कार्बन है, आइसोमरेज. ठीक है, कार्बन का नंबर सेम है. ओके, व्हाट इज द डिफरेंस बेटा? ओनली इन द स्ट्रक्चर. नाउ दिस glyceraldehyde 3 phosphate will be now, yes, we will have 2 glyceraldehyde 3 phosphate, 2 molecules of glyceraldehyde 3 phosphate. Yes, it is a reversible reaction. Now, this 2 glyceraldehyde 3 phosphate will change into, yes, it will change into 1,3 bisphosphate. It will change into 1,3 bisphosphate, right? It will, okay, or I can 1,3 bisphosphoglycerate, right? Yeah, this phosphoglycerate, you already know. Yeah. Everyone is right. So, can anyone tell me which enzyme will be used in conversion of glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate? Jaldi se batao, which enzyme will be used in the conversion of, yeah, in the conversion of glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate? Yes, Rajvardhan, very good beta. It is the hydrogenase enzyme. Already I have discussed it yesterday, and it is glyceraldehyde 3 phosphate dehydrogenase. Glyceraldehyde 3 phosphate dehydrogenase, and I have told you what will be formed: NADH plus H plus. Yes, NAD plus will get converted to NADH plus H plus. Yes, NAD. Yes, Kiran, very good beta. It is, yeah, 1,3 bisphosphoglycerate, 1,3 bisphosphoglycerate. Now, this 1,3 bisphosphoglycerate will change into 3 phosphoglycerate, 3 phosphoglycerate. Again, this is, yeah, again beta, you have to remember that over here, yes, one phosphate will be removed and this phosphate will be added to ADP to generate ADP. This phosphate will be added to ADP to generate ATP. Yes. Now 3 phosphoglycerate will convert into 2 phosphoglycerate. It will convert into 2 phosphoglycerate. You already know 3-phosphoglycerate, the phosphate is present at the third carbon. 2-phosphoglycerate, the phosphate is present at the same second carbon in the same molecule beta. When this type of change is occurring, which enzyme is used? Which enzyme is used beta? Already spoken yesterday. Answer दो इसका फटाफट से सारे. Answer दो. Mutase. Mutase. Very good beta. Mutase. Mutase. Very good. Mutase. Very good. Yes. आशीष, कृति, राजवर्धन, चलो तो थोड़ा बहुत तो याद है तुम लोगों को. Now, from 2-phosphoglycerate beta, what is happening? There is removal of water. There is removal of water, and we will get phosphoenol pyruvate. We will get phosphoenol pyruvate, right? And from phosphoenol pyruvate, we will get pyruvate. Okay, we will get pyruvate. Yes, this enzyme over here, it is enolase. Enolase. And from phosphoenol pyruvate to pyruvate, basically you already know one phosphate is lost, so it will add to ADP to form the ATP. Okay, so yes, over here phosphate is moving from one molecule to another. It is pyruvate kinase. Very good, Ashish. It is pyruvate kinase. It is pyruvate kinase. It is pyruvate kinase. Now this pyruvate beta. Now this pyruvate, this is exactly the same that we have discussed yesterday. इसमें तो किसी को doubt नहीं है. Till now, I hope no one is having any doubt because it is the same thing that we have discussed yesterday. It is the same lecture that we have discussed yesterday in detail. So यहाँ तक पहले मुझे thumbs up दे दो सब लोग. Just give me a thumbs up in the chat box if you have already noticed all the things that we have discussed today were of yesterday's lecture only. Ashish, Ash, Harsh, Rajvardhan, Kriti, very good, very good, very good बेटा. तो अभी आपको यहाँ पे ध्यान देना है. किरण, very good बेटा. Now this pyruvate बेटा, what is happening? Change देखो बेटा यहाँ पे क्या हो रहा है. अब ये जो pyruvate है बेटा, because there is no mitochondria or there is absence of oxygen. अब ये pyruvate mitochondria में जा ही नहीं सकता, mitochondria है ही नहीं. Okay, if we talk about RBC में there is no mitochondria, red blood cells, they are having no mitochondria, so it means that this pyruvate will not be able to enter the mitochondria. Now what will happen to this pyruvate? This pyruvate will convert into lactate. This pyruvate will convert into lactate with the help of enzyme lactate dehydrogenase. Lactate dehydrogenase. This pyruvate will convert into lactate, that is lactate dehydrogenase beta. ठीक है. अब what is happening over here? What is happening over here? Beta, see over here. ये जो NADH plus H plus बना था, this NADH H plus is basically utilized over here. This NADH H plus, it is not giving us any energy because this NADH H plus which is utilized over here, it is again utilized in this reaction. Okay, so this NADH plus H plus, okay, which was formed earlier, will be now again, okay, it will be now used in this reaction and NAD plus will be formed. NAD plus will be formed. So yes, this is again a cycle beta which is going on. Okay, this is again a cycle which is going on over here. This is again a cycle, and this is the only change beta that you have to remember. This is the only change that you have to remember beta, right? This NADH plus H plus which was formed earlier will be now used in this last reaction because there is no oxygen or mitochondria, and ultimately the lactate will be formed. Now beta, coming on to the energetics of this, you already know one ATP is utilized over here. One ATP is utilized over here. Okay, one ATP is utilized over here. So you already know when we talk about energetics, if we talk about energetics beta, energetics. So 2 ATP are utilized. 2 ATP are utilized. How many are formed, beta? How many are formed? Let us count. तो splitting reaction तक there is no formation of ATP. जब split हो रहा है तब तक कोई ATP नहीं बना, because we have already learned yesterday it is called as the preparation phase. When the glucose is getting prepared to get the energy, we are supplying the glucose with energy so that it can generate more energy in future. So till splitting reaction there is no formation of ATP. After that, beta, will this NADH plus give energy? NADH से हमें कितनी एनर्जी मिलती है? How many ATP we get from one molecule of NADH plus H plus? Tell me in the chat box how many ATP we get from one molecule of NADH plus H plus. फटाफट से बताइए चैट बॉक्स में. Yes, 2.5. Ayush, very good बेटा. कृति, very good. हर्ष, राजवर्धन, 2.5 है बेटा. राजवर्धन 2.5 है. अब खुद ही दिमाग लगाओ कि ये दो मॉलिक्यूल्स है ग्लिसरल्डिहाइड के, तो 2 NADH plus H plus बनना चाहिए था, बट ऑब्वियसली ये नहीं बनेगा because this 2 NADH all is utilized, okay, all is utilized in the conversion of lactic from pyruvate to lactate, from the pyruvate to lactate. So ये NADH plus H plus से जो मुझे ATP मिल रहा था aerobic glycolysis में, whatever ATP I was getting in the aerobic glycolysis, I will not be able to get it in anaerobic glycolysis because this is being used. This is again being used. So yes, यहाँ से तो मुझे ATP नहीं मिला. Again बेटा, now, okay, this from 1,3 bis phosphoglycerate to 3 phosphoglycerate, I'm getting one ATP. Okay, I'm getting how many ATPs I'm getting in this reaction, beta, which I'm highlighting? How many ATPs I'm getting in this reaction, which I'm highlighting? Yes, Harsh, very good. बहुत अच्छा दिमाग लगाया बेटा. Obviously बात है ये दो molecules है ना बेटा, क्योंकि dihydroxy has already converted to 2-glyceraldehyde 3-phosphate, right? It has already converted to. Yes, कृति, आशीष, किरण, राजवर्धन, very good. और एक ATP बेटा मुझे यहाँ से मिल रहा है, phosphoenol pyruvate से pyruvate जो बन रहा है. So in totality, how many ATPs are being formed? In totality, how many ATPs are being formed? Yes, 4. Ashish, very good. Harsh, 4. So beta, remember, total ATPs which are formed, total ATPs formed, it is, yeah, 2 plus 2, that is 4. 2 from this reaction, okay, क्योंकि ये दो molecules है, right? And 2 from this reaction, right? 2 from this reaction. So yes, what will be the net ATP, guys? Now calculate the net ATP gain. So yes, net ATP gain in anaerobic glycolysis. Tell me, guys, what is the net ATP gain in anaerobic glycolysis? Yes, beta. Yes, it is 2. Very good, Kriti. Very good, Ashish. It is 4 minus 2 because 2 ATPs are utilized. Total ATPs are 4. It is 2. तो यस बेटा, एरोबिक ग्लाइकोलिसिस में कितना एटीपी बनता है? फटाफट से बता दो, एरोबिक ग्लाइकोलिसिस में कितना एटीपी बनता है? नेट एटीपी कितना है वहां पे? व्हाट इज द नेट एटीपी इन एरोबिक ग्लाइकोलिसिस? दैट इज हाँ, नहीं 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 नहीं, 7 है बेटा आशीष, 7. हाँ, कल बताया था ना 7, 9 माइनस 2 था. टोटल एटीपी 9 और माइनस 2, या 7. क्योंकि ऑब्वियसली बात है NADH का 2.5 काउंट करोगे तो ये 5 हो जाएगा. 2 मॉलिक्यूल्स हो गए तो 5 हो गए. यस, एवरीवन इज राइट, एवरीवन इज राइट. चलिए, वेरी गुड, वेरी गुड. कोमल, वेरी गुड. किरण, वेरी गुड. राजवर्धन, कृति, वेरी गुड. I hope the anaerobic glycolysis is clear to each and everyone. The energetics is also clear. It is the same cycle that we have discussed yesterday, beta. It is just the change in this step. Okay, the change in this step. फटाफट से thumbs up दे दो that you all have done this anaerobic glycolysis. You have beautifully drawn this diagram, this flowchart or this series of reactions in your copy books. Yes, Kiran, Ash, Ashish, Harsh, Kriti, Komal, very good, beta. Perfect, 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 perfect. Now let us now start with the link reaction. The link reaction overview में मैंने बताया था the link reaction. Now my question to you people, link reaction कहाँ पे हो रहा है? where the link reaction is happening. I have discussed in the overview of carbohydrate metabolism where this link reaction is happening. Yes. So write down conversion of pyruvate to acetyl coenzyme A. Yes, very good. Ashish, very good. Kriti, very good. I have already discussed this. Kiran, very good, beta. Very good. You already know, beta, we have pyruvate. Now, if it is like, yeah, if it is an aerobic glycolysis, you already know the product that is formed, it is pyruvate. Okay, the product, yeah, the product that is formed, it is pyruvate. And this pyruvate will now enter this mitochondrial matrix. Okay, this pyruvate will now enter this mitochondrial matrix. Yeah. Hmm. So you already know we have the mitochondrial matrix in which this pyruvate is getting entry. And now this pyruvate, okay, this pyruvate beta, you are already aware. Yes, this pyruvate, it is a 3 carbon compound. Okay, it is a 3 carbon compound and it is getting converted. Okay, it is getting converted into acetyl coenzyme A. It is getting converted into acetyl coenzyme A. And remember, beta, acetyl coenzyme A, it is a 2 carbon compound. It is a 2 carbon compound. Now, it is very much clear that one carbon molecule is lost. One carbon molecule is lost in the form of CO2. जब भी number of carbon कम होता है बेटा, always remember that there is a loss of carbon atom in the form of CO2. Okay, in the form of CO2. In the form of CO2. And remember beta, the enzyme which is responsible for the conversion of pyruvate to acetyl coenzyme A. It is pyruvate, it is pyruvate dehydrogenase, pyruvate dehydrogenase complex. It is pyruvate dehydrogenase complex. Okay, what is happening over here, beta? NAD plus is getting oxidized to NAD plus is getting oxidized to NADH plus H plus. Okay, NAD plus is getting oxidized to NAD. Yes, Ashish, it's right. This pyruvate dehydrogenase. It is getting oxidized to form NADH plus H plus. Now, when I say five, okay, pyruvate dehydrogenase complex beta. Yes, pyruvate dehydrogenase complex beta. Remember that it consists of 5 coenzymes. It consists of 5 coenzymes. Now, the first one beta, the first one that you have to remember, it is lipoic acid. It is lipoic acid. The second one beta, yes, the second one, it is vitamin B1. It is vitamin B1 and it is also called as thymine pyrophosphate. Again, very, very important names. Vitamin B1, it is also called as thymine pyrophosphate. Thymine pyrophosphate, right? Then we have coenzyme A. We have coenzyme A. Okay, that is, yes, that is in the form of, yes, you already know coenzyme A, it is vitamin B5. Okay, it is vitamin B5. It is a B complex vitamin. It is B5 vitamin and B5, it is known as pantothenic acid. It is known as pantothenic acid. You can already see acetyl coenzyme A. So this coenzyme A beta, this coenzyme A, yeah. So this coenzyme A, it is basically the pantothenic acid. right? Pantothenic acid. Then NAD plus beta, NAD plus. We are every time using this NAD plus. Yes, Gamer Alive official, very good. It is NAD plus. Ashish, very good. It is NAD plus. It is vitamin B3 beta. It is vitamin B3. And remember this vitamin B3, it is called as niacin. It is called as niacin or nicotinic acid. It is called as niacin or nicotinic acid. It is called as niacin or nicotinic acid. Then we have FAT. FAT, it is vitamin B2. It is vitamin B2 beta. And this vitamin B2, it is also called as riboflavin. It is also called as riboflavin. Okay. So NAT, that is nicotinic amide dinucleotide, right? So these are all 5 coenzymes that are being used. Yes, yes. So remember, beta, there is loss of CO2. So this reaction, beta, oxidation is taking place. Okay, oxidation is taking place and there is removal of CO2. So this is also termed as oxidative decarboxylation. Oxidative decarboxylation. Oxidative decarboxylation. Okay, there is loss of carbon dioxide also. There is loss of carbon dioxide also. I hope this is clear to each and everyone. The conversion of pyruvate to acetyl coenzyme A. I hope this is clear to each one of you. The conversion of pyruvate to acetyl coenzyme A. चलो फटाफट से एक थम्स अप दे दो बेटा, जल्दी से एक थम्स अप दे दो चैट बॉक्स में. Yeah, just give me a thumbs up in the chat box, beta. चलो हाँ, चलो आशीष, ऐश, हर्ष, किरण, कृति, ऋषि, नमस्कार बेटा. Rajvardhan, very good. I hope this is clear to each one of you. Conversion of pyruvate to acetyl coenzyme A. Conversion of pyruvate to acetyl coenzyme A. Yes. चलो perfect, perfect. Now gamer live official. Okay बेटा, तो अपना असली नाम लेकर आया करो ना, मुझे पता ही नहीं चलता है कौन किस नाम से आ रहा है. कौन किस नाम से आ रहा है, कम से कम नाम ही बता दिया करो असली वाला अपना. लोग क्या होता है कि लाइव सेशंस में मतलब ये तो फिर भी चलो यूट्यूब पे हो रहा है, जूम में ना अपनी शक्लें छुपाते हैं और आई डोंट नो व्हाई, और फिर अभी यहाँ नाम बदल बदल के आते हैं. मतलब आई पर्सनली बिलीव कि नाम अच्छा होगा तो फिर सूरत में क्या रखा है, सीरत मायने रखती है नहीं. ठीक है, अब वो तुम्हारा अपना वो है. सो नाउ वी आर गोइंग टू स्टार्ट विद द क्रेब्स साइकिल. क्रेब्स साइकिल. क्रेब्स साइकिल का दूसरा नाम क्या होता है? व्हाट इज क्रेब्स साइकिल आल्सो नोन एज? चलो फटाफट से बता दो. टीसीए साइकिल. चल, you already know we are going to start with the TCA cycle beta. TCA cycle, or it is also known as the Krebs cycle. Krebs cycle. TCA cycle, it is also called as tricarboxylic. It is also called as tricarboxylic acid cycle. It is also called as the tricarboxylic acid cycle. Okay, it is also known as citric acid cycle. It is also known as citric acid cycle. It is also known as the citric acid cycle. कार्तिकेय चौधरी तुम ही थे क्या वो गेमर वाले? ये तो एक्सपोजर हो गया बिल्कुल. चलो बढ़िया है, बढ़िया बेटा, आया करो. रियलिटी भी मायने रखती है ना. मैं ना I was a doctor on set on one of the reality shows. I will not take the name because it's a live session. I was working as a doctor on the reality show. और तो वहां पे भी ऐसे जो मॉडल्स और एक्टर्स होते हैं ना वो सब थे वहां पे। तो जो रियलिटी फेजेस होते हैं ना वो वहां पे उनको देखने के बाद ही पता चलता है। तो मैंने काफी रियलिटी शोज किए, मतलब एज अ डॉक्टर आई वाज अ डॉक्टर ऑन सेट ओवर देयर, सो यस। So that suddenly reminded of me, when you change names and all that drama and shit. I like just okay. Anyways, चल चल मैं ऑफलाइन क्लास में आऊंगा ना तब मैं तुम्हें बताऊंगा कि मैं जो जिन जिन मॉडल से मिलता हूँ तो उनकी तरफ से जो अपना जो वो दिमाग लगाते हैं, जो मेडिकल खुद ही दवाई लेने लगते हैं, खुद ही डॉक्टर बनने लगते हैं, तो उनके भी मतलब इतने बड़े बड़े मॉडल्स, आई वोंट टेक द नेम अदरवाइज फिर ट्रोलिंग स्टार्ट हो जाएगी। तो ऑफलाइन सेशंस में बताऊंगा। चलो चलो। नाउ नाउ what is the concept, beta? What is the concept? Why there is a need of this cycle? Why there is a need of the cycle? Beta, remember it is final. Yes, it is final common oxidative pathway. Final common oxidative pathway, right? Final common oxidative pathway for macro, yeah, for macronutrients, for macronutrients like carbohydrate, carbohydrates, lipids, and proteins. Now you already know these are the three macronutrients. Yeah, yeah, there are three macronutrients because Makbul, it is I think not a problem from my side because my net is like I have like yeah a very good subscription. So yes. So let us just discuss this bit up. We already know we have the three macronutrients: the carbohydrates, lipids, and proteins. Now these carbohydrates, lipids, and proteins, for them the common oxidative pathway it is the same. That is the TCA cycle. That is the TCA cycle. So you already know if we take carbohydrate beta. अगर मैं carbohydrate लेता हूँ, carbohydrate it is in the form of glucose. It is in the form of glucose. You already know this, right? It is in the form of glucose. Yes, defaulter, I'm teaching it from very basic concept दे रहा हूँ बेटा, पहले concept समझो because why it is needed. डिफॉल्टर तो मुझे खुद ही पता हो गया। अभी देखो कार्बोहाइड्रेट ग्लूकोज है। अब आपको पता है this glucose is breaking down into pyruvate. It is breaking down into pyruvate via aerobic glycolysis. Aerobic glycolysis. And this pyruvate it is forming acetyl coenzyme A. एसिटाइल कोएंजाइम ए। ओके, और इसी एसिटाइल कोएंजाइम में से क्या स्टार्ट होता है? क्रेब्स साइकिल। इसी एसिटाइल कोएंजाइम में से क्या स्टार्ट होता है? क्रेब्स साइकिल, राइट? सो यू नो द मैक्रोन्यूट्रिएंट कार्बोहाइड्रेट इट इज मतलब सारा ब्रेक डाउन करके लास्ट में क्या मिल रहा है? एसिटाइल कोएंजाइम ए। एसिटाइल कोएंजाइम ए। Again, what will happen to acetyl coenzyme A in the mitochondrial matrix? There will be a cycle that is called as Krebs cycle that we will learn, and obviously energy will be generated. Now if I say protein beta, you already know the basic unit of protein, it is amino acids. The basic unit of protein, it is amino acids. Now these amino acids, okay, they will also undergo catabolism. They will also undergo catabolism. They will also break beta, and I will teach in proteins they will also form acetyl coenzyme A. Acetyl coenzyme A. Even lipids, if we talk about lipids, you already know lipids. Basically I'm talking about the fatty acids. The fatty acids. If the fatty acids will undergo beta oxidation, I will teach you all these about when I will be discussing about the lipids and proteins beta. When I will be discussing about the lipids and proteins. Now lipids, these are the fatty acids. Okay, when they undergo beta oxidation, they will again what formed acetyl coenzyme A. So at last, what is formed beta? Acetyl coenzyme A. The all macronutrients are broken down to form acetyl coenzyme A, whether it is carbohydrates, proteins, or lipids. We have already seen in the carbohydrate case how the glucose is getting converted to pyruvate and how pyruvate it is getting converted to acetyl coenzyme A. I have just told you. So this acetyl coenzyme A, now you already know this acetyl coenzyme A will enter the TCA cycle. Okay, it is a part of the TCA cycle. This acetyl coenzyme A in the TCA cycle, it is getting oxidized. It is getting oxidized. Okay, and automatically it is releasing energy. It is releasing energy. It is releasing energy. How we will just see right now. We will just see right now. So I hope this is clear to each and everyone why beta this TCA cycle is important. Why beta this TCA cycle is important. तो फटाफट से एक थम्स अप दे दो बेटा अगर ये समझ में आ गया सबको तो फटाफट से एक थम्स अप दे दो अगर ये समझ आ गया सबको तो यस ऐश वेरी गुड बेटा ऐश वेरी गुड हर्ष वेरी गुड बेटा परफेक्ट 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 बाकी सब यस आशीष परफेक्ट प्रीति वेरी गुड बेटा वेरी गुड बाकी भी एक्चुअली ना रिप्लाई कर दिया करो see no one is sitting in front of me the thing is ki I'm just relying on your response that you are giving on the chat in the chat box otherwise I will इससे बढ़िया तो मैं रिकॉर्डेड लेक्चर डाल देता ठीक है तो लाइफ का मतलब है इंटरेक्शन शुड बी देयर कार्तिक वेरी गुड मकबूल वेरी गुड नाउ बेटा coming on to the site of the TCA cycle, the site or I can say the location. Now TCA cycle, it is occurring in all the organs of the body. It is occurring in all the organs of the body. Now if I ask you the cell organelle beta, can anyone tell me where the TCA cycle is occurring, in which part of the cell? In which part of the cell? Yes आशीष तुम्हें पहले ही पता चल गया था कि सर ये क्वेश्चन जरूर पूछेंगे. So yes, it is the mitochondrial matrix beta. It is the mitochondrial matrix. It is the mitochondrial matrix. Very good beta. So you already know ki you have already, like I've given you an overview beta. Suppose this is the cell membrane. This is the cell membrane, and you already know that the glucose, it is coming through the glucose transporter inside the cell. So yes, this is the glucose. So this glucose beta, it will form pyruvate, right? It will form pyruvate in the cytoplasm, right? I've already discussed this in the cytoplasm. Now this pyruvate will enter, okay, this pyruvate will enter the, yes, mitochondrial membrane. Okay, this will enter the mitochondrial membrane. This pyruvate will form acetyl coenzyme A, and this acetyl coenzyme A will undergo a cycle, okay, and that is something which is called as the Krebs cycle, that I will be discussing right now, that I will be discussing right away. Okay, that I will be discussing right away. Yes, चल इतना क्लियर है. फटाफट से साइकिल स्टार्ट करें फटाफट से एक थम्स अप दे दो that you all are ready for the cycle now, that you all are ready for the cycle. So we'll start with, we have discussed the concept, we have discussed the location, the site, the cell organelle, and we have just given you an overview, yeah, before also. So yes, conversion of pyruvate to acetyl coenzyme done, anaerobic glycolysis done. चल फटाफट से स्टार्ट कर देते हैं. आशीष, प्रीति, ऐश, हर्ष, आशीष, किरण, राउडी, कोमल, कार्तिकेय, वेरी गुड बेटा, वेरी गुड. Now let us now start, and I will give you a trick also beta, how to learn this beautiful cycle in a very like yes, in a very what I would say, in a very easy manner. I will try to make this for you people, right? I will try to make this for you people. Yeah. Now beta, you already know we have acetyl coenzyme A. Try to make this with me beta. Acetyl coenzyme A. Now acetyl coenzyme A, it is a 2-carbon compound. Okay, it is a 2-carbon compound. Okay, now acetyl coenzyme A combines with the oxaloacetate. Okay, it combines with the oxaloacetate, that is a 4 carbon compound that is present in the mitochondrial matrix. That is present in the mitochondrial matrix. That is present in the mitochondrial matrix. Now, what will happen, beta? This acetyl coenzyme A, okay, this acetyl coenzyme A, okay, it will combine with, okay, it will combine with, yeah, acetyl coenzyme A combines with oxaloacetate to form, yeah, it will form one compound beta, and this compound it is called citrate, and this compound it will be citrate, and citrate beta it is a 6 carbon compound. Obviously, acetyl coenzyme it is 2 carbon, oxaloacetate it is 4 carbon. 4 plus 2 क्या हो जाएगा 6. So yes, it is citrate which will be formed. It is citrate which will be formed. Okay, it is citrate which will be formed. Now this citrate it is, now citrate is forming. So the enzyme that will be used over here beta, it is citrate synthase. Okay, citrate synthase जो citrate को बनाने में मदद करे, it is called as citrate synthase beta, citrate synthase. Okay, now this citrate beta, this citrate will be converted into, yes, will be converted into cis, yes, it will be converted into cis aconitate, cis aconitate. So this citrate will be converted into cis aconitate. There will be removal of, yes, cis aconitate, yeah, वो C कर लेना उसको, ठीक है. Now there will be removal of, beta, remember there will be removal of a water molecule. Okay, there will be removal of the water molecule, right? And the enzyme which is used over here, it is aconitase. The enzyme which will be used over here, it is aconitase. Okay, it is aconitase. Okay, so acetyl coenzyme A and oxaloacetate will combine. Okay, 2 plus 4 carbon will combine to form a 6 carbon compound, that is the citrate. Now this citrate, again if there is a loss of water molecule in this, it will form cis aconitate. Now this cis aconitate will again form, yeah, will again form isocitrate. It will again form isocitrate. It will again form isocitrate beta. Now what will happen over here? What will happen over here? Yeah, what will happen over here? There will be addition of water molecule. There will be addition of the water molecule. Yes, पहले स्टेप में वाटर मॉलिक्यूल बाहर निकल गया, दूसरे स्टेप में हमने फिर वाटर मॉलिक्यूल सप्लाई किया. So yes, दूसरे स्टेप में हमने फिर वाटर मॉलिक्यूल सप्लाई किया. Again the enzyme will remain the same. It is cis aconitase. It will cis aconitase, right? It will be cis aconitase. Yes, cis aconitase. Yes, and the compound that will be formed, it will be, yes, yeah, that will be isocitrate. That will be isocitrate. Now, this isocitrate, okay, this isocitrate will form an intermediate product, okay, and that is called as oxalosuccinate. Oxalosuccinate. This is an intermediate product. बेटा कुछ books में नहीं भी दिया हुआ है, so no need to worry about that. This is an intermediate product. Okay, this is an intermediate product. Now this oxalosuccinate will convert into alpha ketoglutarate. Alpha ketoglutarate. Alpha ketoglutarate. Alpha ketoglutarate. Yes. Now, can anyone tell me over here, beta, over here, basically what will happen? Yeah, NAD plus will be oxidized. Okay, over here in this reaction, the NAD plus will be oxidized to form NADH plus H plus. NADH plus H plus. Yes, NADH plus H plus. Yes. Now, over here you already know the isocitrate beta. The isocitrate, it is a 6 carbon compound. It is a 6 carbon compound. It is converting into a 5 carbon compound. It is converting into a 5 carbon compound. It is converting into a 5 carbon compound. What will happen, beta? Yes, very good, Kriti. What will happen? There will be release of CO2. There will be release of CO2. एक कार्बन कम हो गया ना, एक कार्बन कम हो गया, so there will be release of CO2. There will be release of CO2. Yes, and the enzyme over here, since oxalosuccinate is an intermediate product, so you don't have to concentrate over here. Wherever NAD plus and NADH plus is used, yes, Kriti, Ashish, Rowdy, very good, beta. Then the enzyme over here, beta, you already know it is isocitrate dehydrogenase. It is isocitrate dehydrogenase. It is isocitrate dehydrogenase. It is isocitrate dehydrogenase, right? It is isocitrate dehydrogenase from alpha ketoglutarate beta. From alpha ketoglutarate, what will be formed? Succinyl coenzyme A. Succinyl coenzyme A. Again, succinyl coenzyme is a 4 carbon compound beta. What will happen? Again, succinyl coenzyme A is a 4 carbon compound. What will happen? 5 carbon से 4 carbon में जा रहा है बेटा. 5 carbon से 4 carbon में जा रहा है. What will happen बेटा? Tell me. क्या होगा यहाँ पे? चल. There will be a loss of CO2 molecule. There will be loss of one carbon. Yes, CO2. Yeah, CO2 release हो जाएगा. Yes. Again. Yes, again. Now, what will happen, beta? Yeah, the NAD, everyone is right. Ashish Lohar, Harsh, very good, beta. Now, this NAD, yeah, this NAD plus will be converted into NADH plus H plus. Okay, NADH plus H plus. Yes, and the enzyme which will be used over here, beta, you already know it is alpha ketoglutarate, ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. Now my question to you people is, what is this reaction called as? What is the name of this reaction? I am all using it like alpha ketoglutarate dehydrogenase, where CO2 is removed, NAD plus is oxidized, isocitrate dehydrogenase, CO2 is removed, NAD plus is oxidized. What is the name of this reaction? I have already told you about this. Yes, बताओ इस नेम ऑफ द रिएक्शन को क्या बोलते हैं? इट इज ऑक्सीडेटिव. जल्दी से बता दो. इट इज ऑक्सीडेटिव. यस, इट इज ऑक्सीडेटिव डीकार्बोक्सिलेशन. ये तो पता ही है ना सबको, ऑक्सीडेटिव डीकार्बोक्सिलेशन. यस कृति, वेरी गुड बेटा. इट इज द ऑक्सीडेटिव डीकार्बोक्सिलेशन टेकिंग प्लेस एट बोथ द रिएक्शन. ऑक्सीडेटिव डीकार्बोक्सिलेशन. Oxidative decarboxylation. Yes, very good, Rowdy, very good beta. It is oxidative decarboxylation. Now this succinyl coenzyme A will get converted into succinyl, or sorry, it will be getting converted into succinate. It will be getting converted into succinate. Okay, it will be getting converted into succinate. So remember beta, over here, yeah, over here we have an enzyme that will help in the conversion of succinyl coenzyme A to succinate. It is succinate thiokinase. It is succinate thiokinase. Thiokinase. Okay, succinate thiokinase. Obviously, one ATP will be formed. Okay, one ATP will be formed over here. When we are using kinase, the inorganic phosphate will be added. Yeah. So yes, it is, sorry. So yes, this is something which is called as substrate level phosphorylation. This is called as substrate level phosphorylation. Phosphorylation. थोड़ा सा दिमाग लगा के मुझे फटाफट से बता दो, थोड़ा सा दिमाग लगा के बता दो, अभी तक की रिएक्शंस में हाउ मेनी नंबर ऑफ एटीपी मॉलिक्यूल्स आर जनरेटेड, हाउ मेनी नंबर ऑफ एटीपी मॉलिक्यूल्स आर जनरेटेड, यूज योर ब्रेन्स एंड टेल मी. बेटा देखो बेटा, एक NADH प्लस ये रहा 2.5, तो यही हो गया वरुण, 2.5 ये हो गया, एक NADH ये रहा तो 2.5 ये और हो गया, क्योंकि ऑक्सीडेटिव डिकार्बोक्सिलेशन 2 बार हुआ ना, तो 5 तो यही हो गया और एक एटीपी और मिल गया, तो 6 तो मैं दिखाई रहा हूं तुम्हें स्क्रीन पर. हर्ष वेरी गुड, कोमल वेरी गुड, यस किरण वेरी गुड, वरुण समझ आया बेटा, कोई बात नहीं, गलती करो बट सीखो यहाँ पे. एक NADH से, एक NADH से बेटा 2.5 मिलता है, यहाँ पे दो NADH थे, 5 तो यही हो गए और एक ATP 6 हो गया, राइट 6 हो गया. नाउ this succinate, yes, this succinate will get converted into, yeah, fumarate. Okay, this will get converted into fumarate. Now, this is getting converted to fumarate with the help of enzyme succinate dehydrogenase. With the help of enzyme succinate dehydrogenase. Succinate dehydrogenase. And you already know the succinate dehydrogenase. It will be converting FAD to FADH2. Yeah, we have already discussed FAD. Vitamin B2, right? FAD. Yeah, coenzyme. It is converting into FADH2. Yes. One FAD gives how many? One FADH2. How many ATPs it will give, beta? NADH तो 2.5 देता है, FAD वाला कितना देगा? How many ATPs will be released in this? Now will be, yeah, from fumarate. Yes. Yes, Varun Kashyap, very good, beta. Very good. It is 1.5. From fumarate, beta, what will be formed? Remember, we have malate. We have malate, and from malate, what will be formed? Oxaloacetate. Malate से क्या बन जाएगा? Oxaloacetate. So this is a cycle, beta. That's why it is called as the citric acid cycle. Citric acid cycle. Remember, beta, from fumarate, the malate is formed. It is by the help of an enzyme, fumarase. Okay, it is by the help of an enzyme, fumarase, right? And from malate, the oxaloacetate is formed. Okay, over here also the, yeah, over here also what will happen? There will be like, yeah, there will be dehydrogenase acting. Yeah, so there will be NAD converting to NADH plus H plus. Okay, and the enzyme that will be used over here, it is malate dehydrogenase. Malate dehydrogenase. Malate dehydrogenase. Malate dehydrogenase. Yes, everyone is right. Ashish, Kiran, Kriti, very good. Malate dehydrogenase. So again, you can see the oxaloacetate is formed. We have started with oxaloacetate and at last the oxaloacetate is formed, right? Now, before coming to the ATP calculation, if you guys want to remember this, okay, if you want to remember this, so it's a very simple trick, guys. If you want to like, so remember beta, citric cis I, citric cis I और अपने साथ और अपने साथ साथ फूल मलाई और लाई. Now it's a very like those who know Hindi, it will be easier for them to know memorize over here. So citric cis I और अपने साथ साथ फूल मलाई और लाई. Now when I say citric, it's just a rhyming beta. So rhyming है ना तो जल्दी से याद होता है. Rhyming है ना तो पेपर में जल्दी से याद होता है. So yes, it is the citric or I can say the citrate. Cis, you already know it is cis aconitate, citric cis aconitate. I, you already know it is, yes, isocitrate, or O, or you already know we have oxalosuccinate, that is a intermediate product. तो ये और आप भूल भी सकते हो अपना अल्फा कीटोग्लूटारेट, अल्फा कीटोग्लूटारेट, right? We have succinyl coenzyme A, succinyl coenzyme A. We have succinate. Okay, we have succinate. We have fumarate. Okay, malate and oxaloacetate. Oxaloacetate. और लाइन तो भूल जाओ, कभी ऐसा होगा नहीं तुम्हारी जिंदगी में कि कोई सिट्रिक सिस तुम्हारे लिए फूल लेकर आए. सो यस, सिट्रिक सिस आई और अपने साथ साथ फूल मलाई. और लाइक इफ यू like no Hindi, it's way easy. I'll just like, so I hope this is clear बेटा. फटाफट से एक थम्स अप दे दो जल्दी से. I hope this is clear to each one of you. I hope this is clear to each one of you. Now just give me a thumbs up. Yes, Ashish, okay. Ashish says okay, so you are very well aware beta if I'm just talking about this. So yeah, very good. Harsh, Kiran, Varun, Ash, very good beta. You already know we have citrate, citric, cis, I और अपने साथ साथ फूल मलाई और लाइन. So yes, this is how the reaction goes on. This is how the reaction goes on. Yes. डिफॉल्टर अब तो मैंने बहुत इजी तरीका दे दिया समझाने का ना. अब इसमें एटीपी कैलकुलेशन बता दो बेटा. एटीपी बता दो कितने बन रहे हैं. एक तो एनएडी ये हो गया, एक एनएडी ये हो गया, एक एटीपी ये बन गया, और एक NADH प्लस ये, और एक FADH2 ये. तो टोटल NADH कितने बन रहे हैं? नहीं, सब के सब गलत है, बिल्कुल सब गलत है. पहले तो मुझे ये बताओ टोटल NADH कितने बने. चलो इसको अभी लिखते हैं, इसको लिखते हैं फटाफट से. या हाउ मेनी NADH आर देयर? आ रहा बेटा, उसी पे तो कह रहा हूँ. 1.5 बताया था बेटा, 32 बता रहे हैं, बताओ. चलो देखो, पहले तो टोटल एटीपी हमें कैलकुलेट करना है. तो उसके लिए सबसे पहले यू शुड नो NADH कितने बन रहे हैं. NADH बन रहे हैं 3, NADH बन रहे हैं 3. तो यू ऑलरेडी नो 3 इंटू एक NADH से हमें कितना मिलता है. 1 NADH gives 2.5 ATP. It gives 2.5 ATP. So 3 NADH will give 3 into 2.5, that is 7.5 ATP. We are getting from NADH only. Okay, we are getting from NADH only, right? 7.5 ATP बनेगा. Now फिर हमारा आ जाता है FAD वाला पार्ट, FADH2. तो you already know 1 FAD gives how many ATP? 1.5 ATP. And how many FAD? Yeah, how many FADH2 are formed? Only one. Okay, only one is formed. So obviously we have 1.5 ATP. 1.5 ATP, right? We have 1.5 ATP. And the third one beta, the third one, 1 ATP is formed. 1 ATP is formed, right? When succinyl coenzyme A, it is getting converted into succinate. Yes, substrate level for phosphorylation, 1 ATP is formed. 1 ATP is formed. You are very well aware that we have 1 ATP. 1 ATP. Yes, everyone is right. Ashish, Kriti, very good. तो टोटल कितना हो जाएगा? 7.5 plus 1.5, 9 plus 1 कितना हो जाएगा? 10 ATP we will get. 10 ATP we will get. 10 ATP we will get. If you just want, beta, you can see it over here also. You can see from, yes, let me use some other pen. You can see from this NADH beta. From this NADH we are getting 2.5 ATP. Yes, everyone is right. From this NADH we are getting 2.5 ATP. 2.5 ATP. This ATP is like 1 only. Okay. Then we have FADH2. Only one it is formed. It is 1.5 ATP. Okay. And this NADH2, again it is 2.5 ATP. 2.5 ATP. 2.5 plus 1.5, it is 4. 4 plus 1, it is 6. 6 plus 2. Sorry. 4 plus 1, it is 5. 5 plus 2.5, 7.5. And 7.5 plus 2.5, it is 10. So how many ATPs are formed? It is 10 ATP beta. So energetics, I have already discussed the energetics part with you now. Yes, it is the 10 ATP. क्या इतना क्लियर है बेटा सबको? फटाफट से एक थम्स अप दे दो if it is clear to each one of you. फटाफट से एक थम्स अप दे दीजिए if it is clear to each one of you. जल्दी से एक थम्स अप दे दो if it is clear to each one of you. Yes. चलो हर्ष वेरी गुड बेटा. हर्ष वेरी गुड बेटा. Yes, very good. Yes. चलो perfect. Ashish, Ash, Komal, very good. Kriti, very good beta, very good. So I hope the cycle is clear. Site, cell organelle location concept. Now energetics is also done. Now we will study the use of vitamins over here. Okay, in Krebs cycle. Okay, so remember beta, what you have to study over here. Now remember, in Krebs cycle, it was proposed by Hanslet Krebs, so that's why it is called as Krebs cycle. So yes, in Krebs cycle beta, you are very well aware that we have alpha ketoglutarate dehydrogenase, okay, which is undergoing oxidative decarboxylation. Oxidative decarboxylation, right? We have oxidative decarboxylation. So you already know whenever there is oxidative decarboxylation, we have 5 coenzymes that are used, right? We have 5 coenzymes that are used over here. Yes, Kiran, very good, beta. So you already know the first one, it is lipoic acid. Lipoic acid. Okay, we have thymine pyrophosphate. Thymine pyrophosphate. So जहां जहां oxidative decarboxylation होगा, we already know we have like these 5 enzymes that will be used. Vitamin B1. Then we have vitamin B5, that is coenzyme A. Okay, that is the pantothenic acid. Pantothenic acid. Yes, pantothenic acid. Right. We have, yes, NAD plus. You already know NAD plus. It is vitamin B3. Okay. It is niacin. Okay. Or we also call it as nicotinic acid. We also call it as nicotinic acid. Okay. We have FAD. Okay. That is also vitamin B2. Vitamin B2. It is riboflavin. Riboflavin. Right. So yes. Now, now if I ask you the vitamins, okay, which are required in Krebs cycle. So this has been a question, vitamins required in Krebs cycle. It is vitamin B1, vitamin B2, vitamin B3 and vitamin B5. Vitamin B1, B2, B3 and B5. Okay. So we have like these 4 vitamins that are being used over here. Okay. So now I hope this is clear. That is the vitamin coenzymes in Krebs cycle. Vitamin coenzymes in Krebs cycle. Now coming on to the inhibitors. Okay, coming on to the inhibitors. Okay, coming on to the next topic, that is the inhibitors of the Krebs cycle. Inhibitors of TCA cycle. The first one beta, remember the inhibitors of TCA cycle. The first one, remember, it is fluoroacetate. It is fluoroacetate. And remember beta, the fluoroacetate will inhibit an enzyme that is called as aconitase. Okay, it will, yeah, it will inhibit an enzyme that is called as aconitase. Okay, so yes, the second one beta, the second one that we have to remember, it is arsenite. Arsenite. that will inhibit alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. The third one, beta, yeah, the third one, it is malonate, and this malonate will inhibit succinate dehydrogenase. This malonate will inhibit succinate dehydrogenase. Yes, that are the three inhibitors that beta you have to remember. These are the three inhibitors that you have to remember, beta. I hope the energetics is clear. Yes. I hope the inhibitors are clear to each and everyone. I hope the inhibitors are clear to each and everyone. I hope the inhibitors are clear to each and everyone over here right now. Yes. Chalo. Very good. Harsh, Ashish, Harsh. Okay. Kiran, very good. Ash, very good. Very good, beta. Now, beta, coming on to the significance. Okay. Coming on to the significance. Inhibitors I have discussed. The significance, you already know it completely. Okay. It completely oxidizes acetyl coenzyme A. This cycle completely oxidizes acetyl coenzyme A. Okay. Yes. Now it is a truly amphibolic pathway. It is a truly amphibolic pathway. Now when I say truly amphibolic pathway, amphibolic का मतलब होता है बेटा, it is having catabolic role also. It is having catabolic role also. Okay, where the acetyl coenzyme is completely oxidized, and it is also having anabolic role also. Anabolic role also. Anabolic role also. Yes. Yes. Now, when I say catabolic, now, chalo, let me just explain this point. When I say catabolic role, beta, you already know acetyl coenzyme A is oxidized, right? It is oxidized, completely oxidized, complete oxidation होता है, complete oxidation होता है. Now, when I say anabolic role, beta, anabolic role, you already know. For anabolic role, if I just have to explain you, you already know oxaloacetate plus acetyl coenzyme A. I'm just drawing in short, beta. I'm just drawing in short. We have citrate, we have isocitrate, okay. We have alpha ketoglutarate, okay. We have succinyl coenzyme A, okay. Then we have succinate, succinate, fumarate, malate, malate, and again this is oxaloacetate, and then again it is oxaloacetate. So you should remember, beta, when I say anabolic, so it will make something, it will make something. So this citrate will make fatty acid. Okay, it is used for making fatty acid. This is the anabolic role, beta. This is the anabolic role. Okay, the first one. Okay, alpha ketoglutarate, beta. It will form like glutamate. Okay, and again then yeah, it will combine with this and it will form GABA. Okay, succinyl coenzyme A, it is used in heme synthesis, hemoglobin synthesis. That is again a role. Okay, that is again a role. Then you already know oxaloacetate. Okay, it can also form glucose, right? So that is again an anabolic role, an anabolic role. Okay, so yes, that's why it is having catabolic role also, where it is breaking down the acetyl coenzyme A. And again, some of the components of the cycle can again participate in some reactions like heme synthesis, GABA formation, okay, glucose formation, fatty acid formation. So एक तरफ तो ये पूरा तोड़ भी सकता है और दूसरी तरफ ये कुछ कुछ बना भी सकता है, दूसरी तरफ ये कुछ कुछ बना भी सकता है. I hope this is clear to each one of you, beta. Just give me a thumbs up, beta. Yes, just give me a thumbs up in the chat box if it is clear to every one of you. Yes. If it is clear to each one of you, just give me a thumbs up in the chat box. Yes, Ashish, Harsh, very good, beta. Very good, very good. Perfect, 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 beta. Hmm. नाउ कमिंग ऑन टू एनर्जेटिक्स हो गया, इसमें देखो हमने क्या क्या पढ़ लिया, इसमें कंसेप्ट हो गया, ठीक है. वी हैव डन द साइट एंड लोकेशन, द सेल ऑर्गेनेल, ओके. वी हैव डिस्कस्ड ऑल द रिएक्शंस, डन. वी हैव डिस्कस्ड mnemonics to learn this, the energetics done, the vitamin coenzymes done, inhibitors done, significance done. Okay, now coming on to the regulation of TCA cycle. Okay, now coming on to the regulation of TCA cycle. Regulation of TCA cycle. Regulation of TCA cycle. Beta, remember the enzymes that are being used over here. Citrate synthase. Okay. Then we have isocitrate dehydrogenase. Isocitrate dehydrogenase. Alpha ketoglutarate dehydrogenase. Okay. And we have pyruvate dehydrogenase. Pyruvate dehydrogenase. You already know these are like, yes, all dehydrogenases, okay, they are actually favored. Okay, these dehydrogenase, you can see isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase. All dehydrogenase, remember beta, all dehydrogenase, all dehydrogenase will be favored by increased level of calcium. Okay, it is favored by increased level of calcium in the muscle. Okay, increased level of calcium in, okay. Now in brain, TCA cycle is like at pyruvate dehydrogenase, right? Yeah, I don't know who is calling again and again. Happened. Yeah. So remember beta, remember if we have high ATP to ATP ratio, okay, what will happen? The product will be increased. Okay. Now, agar product badega, now beta tell me, if the product will be increased, agar product badega, so it will favor or inhibit the TCA cycle. Yesterday I have given you substrate se product ban raha hai. If the product is increasing, will it favor or will it like inhibit the TCA cycle? जल्दी से बताओ. मैंने कल ये चीज बताई थी तुमको, विल इट फेवर और इनहिबिट. वेरी गुड कृति, वेरी गुड बेटा, वही चीज यहाँ पे यूज करनी है. यस, सो रिमेंबर द प्रोडक्ट इनहिबिट्स द TCA cycle. TCA cycle, even beta high NADH, high NADH or NAD, basically it will also inhibit the TCA cycle. Yes, everyone is right. Varun, Kiran, very good. It will also inhibit the TCA cycle. So yes, isocitrate dehydrogenase, it is like getting inhibited by, okay, NADH. Yes, it is getting inhibited by ATP. Okay. So yes, that is about the regulation of the TCA cycle. That is about the regulation of the TCA cycle. I have discussed the anabolic role of the components of the TCA cycle, right? Inhibitors, significance, vitamin coenzymes, energetics, mnemonics, all the reactions, site, location, cell organelle, concept, conversion of pyruvate to acetyl coenzyme A, and obviously the anaerobic glycolysis. I hope everyone understood that whatever we discussed today. So just give me a thumbs up in the chat box if like you are aware whatever we have discussed today. Yes. Chalo Harsh, very good beta. Very good. Perfect beta. Sure. Sure. Okay beta. Perfect. Kriti, Ash, Kiran, very good beta. Kindly prepare all your notes, everyone, right? Kindly prepare all your notes. Kiran, okay beta, welcome beta, welcome. Sure. ठीक है बेटा. Goodbye.